Hello friends, welcome to the EduSet network. Our today's topic of discussion is DNA transcription, and for this discussion we have with us in the studio our subject expert Dr. Hardeep Kaur, Assistant Professor, Department of Zoology, Ramjas College, University of Delhi. Her teaching experience is 12 years and her specialization is molecular biology. And with this, let us welcome ma'am and request her to begin our today's lecture. Thank you for coming, ma'am.
Thank you, Urvashi. Thanks a lot. Well, friends, today's lecture is on transcription, as already told by Urvashi, and it's a very important process happening, and we will specifically deal with transcription in prokaryotes today, and probably will take up eukaryotic transcription in some other lecture. Now, as you can see on the screen, the central dogma of molecular biology is that DNA undergoes transcription into RNA and RNA undergoes translation into protein. So, the genetic information which is present in an organism is present in the bases present in DNA. This, on being transcribed using RNA polymerase, forms RNA. RNA, which is a single stranded structure, will further be translated into protein. Henceforth, the genetic information present in DNA gets changed into a protein expression. Now, there is a difference in the central dogma whereby the DNA undergoes change, or the DNA further undergoes replication, and the RNA can also change into DNA using an enzyme called reverse transcriptase. This is a difference from the initial central dogma given by Crick. This reverse transcriptase is specifically the characteristic of many RNA viruses. As the name suggests, in RNA viruses, the genetic material happens to be RNA. Now, if a virus, which is an RNA virus, which we call it as a retrovirus, has to get converted into DNA to become double-stranded, it will need an enzyme. This enzyme is reverse transcriptase. This enzyme leads to formation of a double-stranded RNA, rather an RNA with a copy of DNA combined together, which it can integrate into the host genome. Now, after having said about the central dogma, let's come to our primary candidate, that is RNA. RNA, it's a polymer of ribonucleotides. As already said, it is single-stranded structure. The three major types of RNAs are mRNA, transfer RNA, ribosomal RNA. Now, is it only these type of RNA which are present? No, actually, we can call RNA as the genetic RNA and the non-genetic RNA. As already described, I would rather call a genetic RNA as the RNA which acts as a genome. For example, in retroviruses, the RNA present which acts as a genome in a retroviruses would be called a genetic RNA. However, the other three types which I just emphasized, the messenger RNA, transfer RNA and the ribosomal RNA, forms the non-genetic type of RNA. Now, besides this mRNA, tRNA or RNA, there are certain nuclear RNA which I just said could be called a genetic RNA, or microRNA, small interfering RNA, heterogeneous RNAs having different functions which probably we are not interested today. Now, each of them, as I already said, had a specific structure and they perform a specific function. But our concern mainly is mRNA, tRNA and ribosomal RNA. Now, as we are mostly concerned about the transcription, we must emphasize that in transcription, the mRNA and tRNA has a direct role, while rRNA forms the constituent of ribosomes, which are very important part of the translation machinery, which is the next step of transcription, that is from RNA to protein production. Now, as you can see in the chemical structure, there is a ribose sugar to which is attached a base. Here we had given guanine, which is a purine, and along with this ribose sugar, we have a phosphate moiety attached to this. This forms the basic framework of the ribonucleic acid. Now, as again emphasizing what is the difference between an RNA and a DNA. As you can see, RNA is a single-stranded structure, while DNA happens to be a double-stranded structure. If you look at the bases, I just told you that bases, the glucose moiety or the is actually a ribose sugar in an RNA, while it is a deoxyribose sugar in a DNA. Looking at the nucleotides, we have cytosine, guanine, adenine, thymine in case of DNA. In this, guanine and adenine are purines, while cytosine and thymine are pyrimidines. On the reverse, if you see RNA, RNA is a single stranded structure, and the nucleotides which are present or involved in RNA formation are cytosine, guanine, adenine and uracil. That means instead of thymidine or thymine, we have uracil in RNA. Now coming to our main topic, which is transcription. What is transcription? Now we can say transcription is a process involving synthesis of mRNA on DNA template in such a way that mRNA resembles one of the two strands of the parent DNA. Now, as I just told you that transcription is a very important process if we want to decipher the information, the genetic information which is present in DNA and change it into a protein molecule. The middle process is transcription and hence very important. Now the whole genome of DNA need not be replicated, needs to be replicated, but only small portion of the DNA is transcribed. See, the entire, for a replication of a DNA, the entire DNA needs to be replicated, only then it can be passed on to the next cell. For a cell to proliferate into two daughter cells, the DNA replication is a must, and the entire DNA replication is a must. But when I am talking about transcription, we only need a small portion of genome, because probably that portion is required for which the protein synthesis need to be done. So only a small portion of genome is transcribed in response to the development requirement, physiological need and environmental changes. That means probably at a given time, at a given environmental change, a particular protein is required. So only that gene which is present in the DNA, or only that particular portion of the DNA which is coding for that protein would be required to be transcribed, which is very unlike to DNA replication, where the entire genome of DNA is replicated. Now again, the DNA regions that can be transcribed into RNA are generally called structural genes. Now what I had been repeatedly saying that only one strand of DNA undergoes transcription and the other doesn't get. So what exactly is that strand which undergoes transcription? That strand which undergoes transcription of the DNA is the strand which is called the template strand. So template strand is the strand from which RNA is actually transcribed. We also call it as an antisense strand. Just reverse of it, or rather the other strand of the DNA, we'll call it as a coding strand or the sense strand. This is that strand which would be exact copy of the RNA which forms after transcription. That means the coding strand and the RNA which hence is formed after transcription are actually replica of each other. So it is the template strand which acts as the template for the RNA molecule to be transcribed. I hope that is clear. There is one is antisense and one is the sense. Antisense is the template, the sense is the coding. This coding or the sense strand are exact similar to the RNA, while the RNA gets transcribed from the template strand or the antisense strand. Now here is an example. If you see, this is the coding strand GCA, GTA, CAT is GTC, while the template strand is CGT, CAT, GTA, CAG. So after transcription, if you see, it is the RNA which is formed is exact replica of the coding strand. It is GCA, GUA, of course, thymidine has to be replaced with uracil, CAU, GUC. I hope this is clear. Now, if we move on to the mechanism of transcription in prokaryotes, we can actually put this lecture into various headlines like RNA polymerase, promoters, transcription initiation, elongation, termination of transcription. The very first important, RNA polymerase. So, as you can see, as early as 1960 to 61, RNA polymerases were discovered in animals. Plants and bacteria, however, the bacterial enzyme were the first to be studied in great detail. The prokaryotic RNA polymerase, which is of concern to us in today's lecture because we are dealing exclusively with the prokaryotic transcription. This RNA polymerase copies the base sequence of one of the DNA strand. It is an exact copy of the coding strand. That means it takes up information from the template strand, but is an exact copy of the one of the DNA strand, which is the coding strand of the sense strand. A single RNA polymerase is responsible for transcription of all types of RNA. This is complete contrast to what happens in eukaryotic transcription, where actually we have 3 RNA polymerases which codes for different types of RNA. In this case, we have a single RNA polymerase which is responsible for the entire transcription process. It is one of the largest enzymes in the cell and consists of at least 5 subunits. 5 different subunits. In all, there are actually 6 subunits, wherein the alpha subunit is a duplicate. So, the core enzyme has 2 identical alpha subunit and one each of the beta and beta prime and omega. So, together 2 of alpha, 1 of beta prime, 1 of omega together constitute the core RNA polymerase, prokaryotic RNA polymerase, whose size is around 400 kilodaltons. However, with the binding of another factor, which is a very, very important factor for a prokaryotic RNA polymerase, that is a sigma factor, the complete polymerase, which is now called the holoenzyme, is formed and is around 450 kilodaltons in size. So, here you can see this, the rough depiction of prokaryotic RNA polymerase, where you have 2 alpha units, 1 beta prime, 1 omega. This forms the core enzyme. Now, if we consider sigma factor along with it, we get a holoenzyme. So, core enzyme plus the sigma factor gives a holoenzyme. So, as I have shown here, the 2 largest subunits, that is beta and beta prime of the core enzyme, are actually involved in catalysis, while alpha 2 and omega helps in the polymerase assembly and regulation. Now, the sigma unit is extremely important factor which is present in RNA polymerase. The core enzyme is not a specific enzyme. The specificity of the RNA polymerase is only achieved once the sigma factor combines with the prokaryotic RNA polymerase. So, as already said, the holoenzyme, that is 2 of alpha, 1 beta prime, 1 omega and sigma, that is called the holoenzyme, is a highly specific enzyme for immediate early genes, while the core enzyme, as already said, do not have much specificity. The core enzyme, however, is capable of transcribing both DNA strands. Now, if we go on to understand the function of the sigma factor, I have been repeatedly telling you that sigma factor is extremely important. So, what are the functions of a sigma factor? The sigma factor helps in recognition of the start signal on the DNA molecule, and it directs the RNA in selecting the initiation site. In fact, it was called sigma from a Greek letter which specifically stands for specificity. So, the specificity of prokaryotic RNA polymerase, again I emphasize, is achieved due to the binding of the sigma factor to the core enzyme. Now, again, there are only 30% of sigma factors present per as compared to, if we consider core enzymes and as 100%, only 30% of sigma factors are present. So, at any given time, the holoenzyme which is present is only 30% or 1/3 of the total core enzymes present, because there are only 30% of sigma factor. So, after initiation of prokaryotic transcription, the sigma factor dissociates from the holoenzyme, leaving the core enzyme. After giving it the right direction, it goes off to the another core enzyme to start transcription for another core enzyme from the promoter region. So, most common equalized sigma factor is sigma 70. We also have other sigma factors like sigma 32, sigma 41.5, sigma 28, etc. These sigma factors are responsible for transcription only certain adverse conditions. For example, some temperature changes, nitrogen deficiency, certain adverse conditions, other sigma factors become important. But otherwise, the most important E. coli sigma factor is sigma 70. Now, as you can see in this slide, this is the E. coli sigma 70 promoter. As you can see, the RNA polymerase binds to specific initiation site upstream from the transcribed sequence, which are called promoters. So, we are now discussing the E. coli promoter, and since it is specifically dealing with sigma 70 factor, so we call it as a sigma 70 promoter. Now, if we see the E. coli promoter, if we consider +1 as the transcription start site from where the RNA synthesis begins. So, upstream region to this transcription start site we denote it by minus sign. So, two regions are extremely important, which is -10 region and -35 region upstream of the transcription start site. Now, the sigma 70 promoter consists of a sequence of about 40 to 60 base pair. The entire promoter region before the transcription start site is about 40 to 60 base pair, which is defining our transcription initiation and is of utmost importance. So, actually the polymerase binds from about -55 upstream to +20 downstream of the transcription start site. That is the region where exactly the RNA polymerase in prokaryotes binds. Now, as I just told you in the previous slide, that this -10 region and -35 region is very, very important for the binding of RNA polymerase, for the right binding of RNA polymerase. Then let us consider this region one by one. The -10 sequence in the E. coli sigma 70 promoter, it was first recognized by Pribno in the year 1975, hence called Pribno box. This sequence is centered at about -10 position with respect to the transcription start site. As I just told you, we are considering the transcription start site as +1. The consensus sequence which is present here at -10 position, which is we call it as a Pribno box, is a 6 base pair region which has which can be defined by nucleotides TAT, that is TAT. Out of this, the first nucleotides TA and the last nucleotide T are highly conserved among all prokaryotes. This is the place where DNA unwinding begins with the help of polymerase. Now, I told you about another sequence which is -35 sequence. Again, it is a very conserved sequence. It is a hexamer of nucleotide TTGACA. It enhances the recognition and interaction with the polymerase sigma factor. That means the entire, see, if it is a core enzyme do not have specificity. It can bind to any position on a DNA molecule. But if the sigma factor is attached to the core enzyme to form a holo enzyme, it provides specificity. The entire polymerase can occupy a position between -55 to +20 region. Now, while occupying this position, the sigma factor has direct interaction with the -35 sequence, and because of this gives specificity that yes, the holo enzyme is right now present at the right position. Now, -10 is the position where unwinding of the double stranded DNA takes place. As already said, +1 is the region where transcription begins. Now, here I will just show you, this is a transcription start site which I said +1. It's in most of the cases it is a purine, with guanine being much more common than adenine. Now, if you see that if we consider this as a double stranded DNA, the +1 is the transcription start site. As already said, it could be guanine, it could be adenine. Most common is guanine. This is the place where start of transcription occurs. Now, -10 region, that is the Pribno box, that is TA-AT region, where I again told you the TA, the initial TA and the last TA is highly conserved among all prokaryotes. At -35 region, we have TTGACA, which is again a conserved region which tells the holoenzyme yes that it is at its right position. Now, another difference from DNA replication here is that there is no need for a primer in this case. Transcription does not require any primer. So, no primer is needed for RNA synthesis. Now, coming to the main process of transcription, that is transcription initiation, elongation and termination of transcription. We will deal with this one by one. Coming first to the transcription initiation, as you can see in this slide, there is a RNA polymerase binding to the promoter region. This leads to the formation of a closed promoter complex. It is a ternary complex, it is not a ternary, it is a binary complex, it is a closed complex where the DNA is still in an unwind manner. It is just a polymerase binding. The entire polymerase is there, as you can see the two units of alpha units, one beta, beta prime, omega as well as sigma 70 which is present. The entire holoenzyme is there. It is binding at a position upstream of the transcription start site in the promoter region. The sigma has a direct influence with the -35 sequence and hence defining specificity. -10 is a position where the unwinding will begin after initiation. So, as you can see, there is binding of promoter, the holoenzyme is binding to promoter site. There is a formation of a closed promoter complex. We call it as closed promoter complex because the DNA is still in an unwound formation. The holoenzyme then slides along the DNA and finds the double stranded -35 and -10 DNA sequences. Now, once it reaches, as you can see in the next slide, that is initiation site. Once it reaches the -10 position, the unwinding of DNA from double to single strand takes place. Now, with the unwinding of DNA, the process leads to formation of an open promoter complex. Now, we call it as an open promoter complex because now the polymerase is binding to the open DNA. DNA is no longer in a double stranded manner. Now, as we go further, as I said, there is unwinding of DNA and separation of DNA to form open complex. Now, after that there is an initiation of RNA chain. After unwinding, there is an incorporation of nucleotides up to 9 bases. Now, most importantly, this RNA remains there for some time, and for example, there is a 1 to 2 second of time difference before the initiation further progresses. During this 1 to 2 seconds, there is a formation of 9 bases. We must understand not only the promoter, the next probably 20 to 50 bases in the DNA also defines the transcription. So, the first 9 bases of the RNA which are formed are very important. They define whether the transcription taking place is okay or whether it is not fine. This is called abortive initiation. If the RNA polymerase is taking more time, let us say more than 1 or 2 seconds to form these 9 bases, then it leads to abortion of this transcription. The transcription just stops. It does not progress further. This is a very important regulatory event. Now, if everything goes fine, the 9 nucleotides hence formed are perfectly formed in time, the RNA polymerase is acting perfectly fine, then the transcription keeps on happening. Otherwise, it leads to abortive initiation. So, for any particular transcription, we might get many small oligonucleotides of 9 bases long. Furthermore, as you can see in the slide, the next process which takes place after initiation is transcription elongation. Now, after the initiation has happened, as you can see in this slide, after initiation the sigma factor comes off the holoenzyme and what is left is a core enzyme. The core enzyme or the holoenzyme has already given specificity to the DNA, to the RNA transcription. It has defined that this is the right place where the transcription should have started. Once its work is done, the sigma factor comes out of the holoenzyme, Though core polymerase is left behind, the sigma factor is now free to combine with other core polymerases and start transcription again from the promoter region, probably from the same promoter region or from some other promoter region of another structural gene. So, as I defined you initially that there is only 30% sigma factors which are present at any given time in a prokaryotic cell. Now, after transcription initiation, the next process is transcription elongation. Again, as you can see in this slide, as elongation proceeds, there is, as RNA polymerase keeps on moving further, there is an unwinding of DNA. You will see that there is a formation of transcription bubble. There is unwound DNA present, there is an RNA polymerase. This region is about 55 nucleotides long, 17 base pair long, which we call it as a transcription bubble. Now, this is a region which, as I told you, transcription bubble of 17 base pair long, which forms a ternary elongation complex. Ternary, as the name suggests, it has got 3 components. It has got DNA, it has got a nascent, nascent means newly formed RNA, and it has got RNA polymerase. Together it forms a ternary elongation complex. This ternary elongation complex, as you will see in the slide, behind this bubble, transcription bubble, the DNA which was unwound before is now undergoing rewinding, while the DNA ahead of this bubble is undergoing unwinding. So, behind it is rewinding, in front it is unwinding in anticipation of the RNA polymerase. So, as you can read it, the enzyme releases the sigma factor. It forms a ternary elongation complex of polymerase, DNA and the nascent RNA. The transcription bubble, which is around 17 base pair long, it moves along DNA with the polymerase. The polymerase moves at an average rate of 40 bases per second at 37 degrees Celsius. That means in 1 second it is covering 40 bases. However, this is not specific for every polymerase. In case of certain viruses like T3 and T7 polymerases, this rate is much higher. It is around 200 bases per second at 37 degrees Celsius. Now, following transcription elongation, the last process which happens is transcription termination. Now, as you can see in this slide, the DNA strand has rewound as the RNA polymerase is moving further, while in further the important part is the transcription is again happening in the pi 5 prime 3 prime direction. Now, you can see that a nascent mRNA is formed. Now, once the complete gene or the DNA or the gene has transcribed into an mRNA, the structural gene has undergone transcription, it so happens that the polymerase transcribes a region on mRNA which we call it as a hairpin region. This particular termination which occurs, we call it as a row independent termination. The RNA polymerase continues transcription unless it reaches a terminator sequence which we call it as a stop signal. The stop signal is a GC rich region which is a hairpin structure. After having transcribed a hairpin structure which has a stem and a loop, the RNA polymerase takes a pause. After that, it will come across certain U residues. After synthesis of this hairpin RNA, the U residues in the RNA base, they undergo weak pairing with the A residues in the antisense DNA. This is a signal which says that termination has to take place. Due to this weak binding of the U residues in the RNA with the corresponding A residues in the DNA, there is a removal of RNA polymerase from the DNA. This dissociation of the core enzyme from the DNA leads to termination of transcription. This is called row independent termination. Again, we must notice that the termination signal is actually present on mRNA which is newly synthesized. It is the hairpin loop followed by the poly U site which defines the stop signal. There is another kind of transcription termination, which we call it as a row dependent termination. Here an accessory factor called the rho protein, which is an hexameric protein, it mediates transcription termination. Rho, as I just said, is a hexameric protein. It hydrolyzes ATP in the presence of single stranded RNA. It binds to a stretch of 72 nucleotides in the nascent RNA. The newly formed RNA, the rho moves, here you can see in this figure the green hexameric protein is present, which is present on the nascent RNA, is the rho protein. It moves along, it actually binds to a stretch of about 72 nucleotides on the newly formed RNA. It moves along the RNA, and as was the case in case of rho independent termination, the signals are recognized in the newly synthesized RNA by the rho protein rather than the template DNA. It reaches a place. Now the signal could be again a hairpin loop, or it could be something else. We do not know too much about it, but there is a stop signal which is identified by the rho protein while moving on the newly formed RNA chain. After moving on the newly formed RNA chain, it comes across this signal, it leads to termination, and the RNA polymerase or the core enzyme just comes off the double stranded DNA molecule and a newly synthesized RNA is formed. Now, unlike in eukaryotes, which has a nuclear envelope, in case of prokaryotes we do not have any such membrane. So, because of that, this newly formed nascent RNA can immediately be translated while it is undergoing translation. So, ribosomes can actually bind to this newly formed RNA and start the process of translation while the transcription is not yet been completed, which is very unlike eukaryotes, where the transcription has to first finish, it has to come out, and then translation will occur in the cytoplasm. Now, here I am emphasizing a drug which is a therapeutic drug, which is a rifampicin, which is a tuberculosis, which is used in tuberculosis treatment. This drug specifically binds to the beta subunit of RNA polymerase, hence inhibit the RNA synthesis in the mycobacterium. Hence, rifampicin is the drug of choice for tuberculosis treatment. The RNA pol of other prokaryotic system is also similar to that of the E. coli in structure as well as in the function. Some of the interesting facts of RNA transcription, of the transcription in prokaryotes, that in 1959 Severo Ochoa won a Nobel Prize in Physiology or Medicine. He developed a process for synthesizing RNA in vitro using polynucleotide phosphorylase. In 2006, Roger Kornberg won Nobel Prize in Chemistry. He was awarded for his studies of the molecular basis of eukaryotic transcription, however. Now, here is one of the video where you can see the RNA polymerase binding. It is leading to elongation. I will just repeat it one more time. There is a transcription, I will just come back to it. There is a transcription start site, there is polymerase, the polymerase is binding to the template strand leading to elongation, coming up to the end sequence, and finally termination of transcription taking place. With that, I end my lecture. This is all I wanted to tell about the prokaryotic transcription. Thank you.
Thank you so much, ma'am, for covering the topic in such a lucid manner and also with the wonderful illustrations for our students, so they get also an idea of how the phenomena occurs. And with this, we have come to the close of the lecture. So we shall thank ma'am for being here and talking to us and sparing her valuable time. Thank you for watching. Have a wonderful day, and thank you so much, ma'am.
Thank you so much.